So I think although many of you will be familiar with DNA sequencing, I thought it would be best to start this workshop with just a brief overview so everybody knows where everybody stands. I should say we are using this hashtag #OhNo through the conference, so feel free to tweet to your heart's content. And the idea of this is to just give a bit of a foundation for the analysis of sequence data that we'll be looking at a bit later. So the question really is for everybody who starts working in bioinformatics, I think, is what makes sequencing so hard? So I think there's lots of different factors that can lead to people struggling with sequencing or it being a bit of a daunting task. One of them, which happens at a very early stage often, is that not all tissues are equal. So there can be things which are much harder to extract DNA from than others. So for example, this azellus in the top left corner here, they have very hard exoskeletons that they can be very difficult to digest. Not all organisms will give up their DNA so easily. So actually things like this mangrove here, and often the case in plants and other fatty organisms, their tissues can have these metabolites and other inhibitors, which actually just make extraction chemistry very challenging. Another thing, like with this azellus, is that there might not always be a lot of tissue. So we've seen through the Slack channel what some of you work on. Some of them are very big things, but some of them are also very small. And obviously having enough DNA to answer the questions that you want to answer is not always super easy. And the reality still is that we really can't sequence chromosomes end to end. So there has to be some kind of shearing, and we have to make some decisions about which bits of DNA we're actually able to sequence. And the thing that it always comes down to, at least in part, is that the whole process costs money, and we don't all have unlimited amounts of money. But over the last few years, we have seen a big progression in DNA sequencing. So the Human Genome Project, which took place from 1990 to 2003, was the job of a lot of people and cost around $1 billion, it's estimated. I think this is actually, on the right here, this is a book from the Wellcome Collection where they printed off the first draft of the human genome. And it's not a brag, but rather to show the difference in sort of 20 years or 30 years can make, which is that during my PhD, I actually assembled a genome about the same size as the human genome and analyzed hundreds of whitefish whole genomes. And I think that's the reality for many of you looking at the projects that you're doing. So this progression is obviously happening quite quickly. And that's being helped by the fact that actually sequencing is getting a lot cheaper. So here's a curve from the NIH. And you can see that there's this, well, in 2008, 2009, there's this very steep drop off in price. And although it's still reducing, it's kind of leveling off slightly. And other people clearly think that sequencing is a good idea and appreciate this cost because companies like Illumina and Pacbio, their stock prices are going up and people are making very big investments in sequencing technology. So Pacbio, for example, if you invested early, you've done very well because they had this almost $1 billion investment earlier this year. And these technologies are being used more and more as we sort of go towards things like personalized medicine. These costs are dropping. And most of us, I think, who don't work on humans are having the sort of residual benefit. So although the improvement in the drop in price is not as drastic as it once was, I think there still is a drop in price. And I think competition between at least these two companies is still going on. Although they did almost merge a couple of years ago. So who knows what will happen in the near future. One of the other things which has led to this big drop in price is the increased output of sequencing. So if you were doing sequencing 5-10 years ago, you were limited to very small outputs. So these on the left here are the very old Illumina machines, which would output about a gigabyte of data. And now if you're working on these new Novosec machines, you're getting about 6 terabytes of output, or you can get up to 6 terabytes. So that's 20 billion reads. And to be honest, this scale is pretty remarkable and it's ever increasing. Now, there's obviously a lot of jargon associated with sequencing. So although some of you might have done some approaches, you might come across words that you're not familiar with or phrases. So these might include high throughput sequencing, next generation sequencing, second generation sequencing, short read sequencing, third generation sequencing, long read sequencing, and then these sort of brand names, Illumina, Pacbio, Nanopore. But really what these can be broken down to is the fact that high throughput sequencing is next generation sequencing. And then these sort of sub-generations refer to the type of technology that you're using. So second generation typically refers to short read sequencing, which is often done with Illumina. And third generation is now this longer read sequencing. So that's normally things like Pacbio or Oxford Nanopore data. So to understand really how today's sequencing is going, it's maybe good to look back and see how people did it, not in the not so distant past. So one of these ways was Sanger sequencing, which some of you might have even been doing at the beginning of your careers. And the way Sanger sequencing works is you basically take your DNA that you want, which has already been cut up to some extent, you attach these different colored nucleotides, which go on here, and then you run them through this capillary gel, which basically pulls your DNA strand through, and a laser basically measures the fluorescence that comes off these different labeled nucleotides. So you end up with a graph like this. If you've worked with Sanger data, you'll know the pain of working with these. This is a very nice one, and they often didn't look this nice. And although Sanger was a very powerful technology at the time, one of the challenges was you'd often get failed runs, and you would constantly have to rerun plates and plates of samples. So that was a very painful thing, and complex samples with more inhibitors and things like that can also suffer quite badly if this reaction doesn't work as well as you hope. Now, a lot of the other technologies actually use a similar approach, which is this sequencing by synthesis, and Illumina is one of the ones who does that. So this is an Illumina sequencing flow cell, which is from the new Novasec machine. I don't know how many of you have actually seen one of these, but actually I've got here, I think it's a MySeq. So from back in the day, this is a small MySeq cell that you would use in a MySeq machine, and you can see that the difference in scale is pretty remarkable. So this is the size of like a stamp, if you can't see it. And Illumina is pretty amazing because what it does is you have a flow cell like this, which basically has this, this textured matrix on here, which they're always improving. And that matrix basically has these different oligonucleotides attached to it. And so what happens is your DNA fragments that you've already extracted and cut up will bind to these oligonucleotides with the Illumina primers that you've attached. DNA then is folded over, and you basically synthesize DNA, this complementary strand, as it goes. You then end up with these two strands, and then in some protocols, basically this reverse strand is washed off. But what ends up happening is you've essentially grown a small patch of that sequence around the area. So from one initial DNA strand, you've basically synthesized a big clump of it. And then what the machine does is it actually uses, again, this fluorescing approach where you attach nucleotides that have these different fluorescents. And what the machine does is it basically captures what nucleotide you've actually put in there per cycle based on the illuminated flash. So one of the things which Illumina has done recently, which we can get into another time, is they've changed from a 4-channel chemistry to a 2-channel chemistry. So in previous iterations of Illumina technology, things like MySeq, I think, and NextSec maybe, they use this 4-channel chemistry where each nucleotide has its own color. So in this example, A's are red, G's are blue, T's are green, and C's are yellow. And that's great. Until they decided that to save money and throughput, they could change to this 2-channel chemistry where different combinations of light confer nucleotides. So A is signified by green and red being emitted, T is by green, C is red, and G is no emitted light. Those of you who've worked with data might already see the problem here, which is that if your machine doesn't work, or you get a round which doesn't work, or a cycle which doesn't work, I should say, you will end up with a G call in your Novasec data, which is not a real G call. It could have been anything. So although this technology was designed to increase throughput, there are some drawbacks. And so if you're combining data across platforms, this is especially important to bear in mind because one G call does not equal another across your samples. But essentially what Illumina managed to do is they have these patches of sequence, and the patch basically allows the machine to pick up that fluorescence in a much higher quantity. So it's not a single molecule that's emitting this photons, but it's actually this whole clump, which allows them to more accurately call the nucleotide for you. Some of you are working with Pacbio data. Pacbio machines are also evolving quite rapidly, and there's new ones coming out, it seems, every year or so. Right here is an example of Pacbio's latest technology, the Hi-Fi system. And some of you might still have this, if you don't work with Pacbio data, some of you might think that, well, Pacbio data is still very error prone. But the reality is Pacbio have generated this new Hi-Fi system, which is pretty remarkable and has similar accuracy to Illumina reads. So that trade-off that you used to have of length over sequence quality is getting smaller and smaller. So the Pacbio system works slightly differently, where you actually take your double-stranded DNA and you ligate these two different ends on it. This is what they call this smart bell technology. And basically what they do is this then sits in a sort of well on a plate, and the sequencing bit, which is making the fluorescence happen, is kind of whizzing around this molecule in a circle. And so you can see in this diagram here, you just go around and around and around your circularized molecule. And what it does is it allows you to actually correct the small errors that there would normally have been. So I think in old Pacbio data, you would end up with these errors. And now you kind of are able to call this consensus read by just going over the same circularized bit of DNA many times. Then there's Nanopore, which is, I would say, one of the most fashionable sequencing technologies nowadays. And their branding has worked very well. So Nanopore have this whole host of different sequencing machines, which were, I think, initially designed to help you use them in the field or in conditions where you might not be able to house, say, a whole Pacbio machine. They're relatively far more affordable than the other technologies in terms of the machines. And you can see some of them are small enough to plug into your phone. And some of them here are massive and produce massive amounts of data. Nanopore reads are much longer than both of the other technologies. They can be up to hundreds of kilobase. And the way Nanopore works is slightly different to the others. So it's not sequencing by synthesis, but what you do is you take your DNA, you kind of rip the two strands apart, and you pull one of them through this modified, I think it's a bacterial cell wall kind of pore membrane. And what the machine actually does is it measures the voltage across this membrane. And so each nucleotide, because of its different chemistry, will change the voltage slightly differently. So you can see in this diagram here. And from the voltage running across this pore at any one time, you're able to actually call the bases. And I believe that this technology also benefits from the circularization, where you can actually run a molecule through many, many times, which is how you get the improved accuracy of nucleotide calling. So maybe it's time to think about what most of us are doing, which is some kind of whole genome shotgun sequencing. I won't be talking much about reduced representation sequencing because in this workshop, many of those data sets are not so applicable. And I think this is actually becoming much more common. But the reality is, as I said earlier, we can't sequence end to end. And so what we typically do is we cut up the DNA into smaller pieces. We're then able to generate many millions of reads from these many millions of reads from these different fragments. And what we often do later then is we align them to some kind of reference, or we create some kind of de novo assemblies, which we use to call genotypes or understand something about structure. Or in the case of this workshop, we use the fragments and the sequences themselves to understand something about the genome. Now, one of the important things I think with sequencing projects, which is not often iterated enough, is how important project planning is. So if any of you haven't started your project yet, this is something worth bearing in mind. But there is this trade-off of the number of sites you want to sequence, the number of samples you have, and the depth of sequencing. And I actually think this question is becoming, in a way, more open and slightly more challenging to address, just because of the number of different options that are available. So you might, for example, need to get a population level overview of your system, and then you might be willing to trade off some depth in exchange for the number of samples. If, however, you want to do something like structural variant calling, the depth is obviously critical to being able to understand something about those structural variants. And so you might be faced with this trade-off. And it comes back essentially to what I said at the beginning, which is why sequencing is difficult. So it's important to identify your question, think about your budget, which may be you or your boss's responsibility. You can even think about things like, do you have a reference genome? Does it make sense to be sequencing hundreds and hundreds of whole genomes if you don't have a reference? It might make sense to think about how big your genome is. Obviously, the output of these machines is constrained by basically the number of bases you want to sequence. So if you work on a 100 gigabase genome, you are going to get much less population level sampling for the same amount of money than if you work on something with a very small genome. DNA that you can extract is actually, I think, a very real consideration for people that work on small things. So, what's the realistic depth that you're going to get from a single individual if you have a limited DNA to start with? And how easily can you get that DNA? Are you going to end up with months in the lab trying to produce a new extraction protocol? Or are you better off just doing some kind of different sequencing approach? And here's a table kind of just summarizing what we've said. So, you have this short read data, which has very high accuracy, can be used for many different things. So things like rad sequencing, whole genome, Hi-Fi for genome assembly and scaffolding. Sorry, not Hi-Fi, Hi-C. So those of you who are not familiar with Hi-C allows you to understand the physical linkage of different markers, because you basically bind together bits of DNA before you cut it up, so you have some information about their position. And then you have these long read technologies, which we said. So much longer reads, traditionally lower quality, although that's improving very rapidly. And these are typically used for things like genome assembly or understanding structural variation. So here's kind of the main link between what I've just said and what we'll be doing in a little bit, which is the Bash refresher course. So what does my data look like? More often than not, you will end up with a file like this from the sequencing machine or from your sequencing center. This has obviously had some stuff done to it first. The machine doesn't just spout this out, but typically you will get it in this format. And what you end up with is, at the top here, you have this header line, which contains the machine information, normally some run information, and whether your read is like a forward or reverse strand is also often kept here. So I think in this case, this is what this one is for. And that header will always start with an @. This is a Fastq file. And then you'll have your sequence. Then you will have a plus, and then you will have a string that is your quality score. So this is, in this example here, it's just letters, but there is a whole host of symbols. It uses the whole sort of ASCII character library that's available. And each of these different characters here corresponds to a Phred score. What a Phred score is the probability that that base call is wrong. So if you there are tables you can look up to see the conversion. Phred scores are also a bit of an enigma in the sense that they don't always make sense to compare Phred scores across runs, but they can be very useful within a sequencing run to figure out if you have some badly sequenced strands. And this is obviously one read. So in your Fastq file, you will end up with many millions of these probably. And as you have seen, each of these is basically 4 lines. So it's a bit compressed here, but in reality, you have the header line, the sequence line, the plus, and then the quality line. So that's just worth bearing in mind when you're trying to figure out like how much data you have and things like that. Now, one of the things we'll be going over in the tutorial in a bit is using Fastp. So Fastp is a quality control or a quality checking software, which I think is very useful. Some of you might be familiar with FastQC, which I think used to be sort of the gold standard, but I actually think with new data, especially from things like Novosec, Fastp has a lot of benefits, which I hope you'll see. And so I thought I would just talk through kind of the expected output of Fastp so that when we get there, you can maybe interpret it a little bit for yourselves. Fastp, when you give it a forward and reverse file of sequencing data, will tell you everything you need to know about it. It will give you far more information than you'll probably ever use, but I think it can be particularly useful if you start to notice strange patterns in your sequencing data, which if you work in sequencing data for long enough will happen. And for that reason, I think it's a bit better than FastQC, which sometimes highlights patterns that aren't real and sometimes misses things which are. Fastp will give you this general overview of your sequence information, things like mean read length. It will give you the insert size peak, so what's the mean distance between your reads in your paired end library. It will tell you the kind of sequencing that you did, in case you've forgotten at this point. And it will give you some overview of what the quality of these bases look like, and it will even tell you how much it improved it. So here it's actually run on a I think it's an already filtered data set, but you can see there's still a marginal increase in things like the number of high quality bases. Here it's like a very, very small increase. And it will tell you what you basically end up with at the end and how many reads it throws out if you give it some kind of conditions. It will also tell you, which can be especially useful if you have any adapter left in your sequences. So some of you might get sequence back from a cluster, a sequencing center or consortium, and you don't actually know what you've been left with. Have they cut off the adapters? Have they demultiplexed for you? And Fastp should help you get at all of those questions. And then it will give you some nice graphs like this. So it will tell you, is there a particular place along each read where the quality dips? So somebody might have bumped the machine, or there might have been a problem with some of your reagents in that step if they weren't put into the flow cell correctly. Here you can see there's this big dip around, I don't know, 23. And this is obviously really good to know if you want to do any processing later on, although with things like this, I would leave it. But especially at the ends of the reads, it can help you know if you need to trim off the ends, for example. And it will also show you a graph like this, which shows along the position of the read, do you have kind of a bias in the base content? So this comes back to what I was saying about this G issue based on this 2-channel chemistry from Illumina. And what you actually see if this G issue applies to you is you'll see a massive increase in the number of Gs called at the end of a read. So obviously this difference here is like GC bias. That's what causes the 2, but at the end, you see they all start going a bit crazy. And what you'll see if your data is highly affected is that this G, the blue line in this case, will actually just start spiking massively. And that might tell you that you need to trim off those poly G tails. And Fastp actually will tell you if that's a problem, and it will detect if your data was collected with Novosec data. And there are settings to remove those poly G tails. It will also output a matrix like this, which actually links a little bit to this workshop. So this is a K-mer counting matrix. And here it uses this color gradation system to show under and overrepresented K-mers. So the darker the color, the more overrepresented. You can see here this TTTTT is very abundant. Same with the poly A. But this one down here, for example, is lower than expected. And this, again, if you have this poly G issue, this lower corner here typically is like completely black, which is really easy to tell. And it produces one of these for pre-filtering and also post-filtering. So if it is a problem, you should be able to see how well you've been able to fix that problem during the filtering as well. So I think that's just a bit of a whistle-stop tour of what we're going to go through.